Welcome. Today we will discuss fertilization in domestic animals. So fertilization simply it is a fusion of male and female gamete resulted formation of zygote. And this fertilization takes place at ampulla, the part of the oviduct. So let's see uh, how the spermatozoa reach to the site of fertilization. For fertilization, the spermatozoa have to reach at the site of fertilization, that is ampulla. So the spermatozoas are deposited. The semen is deposited at vagina in most of the domestic animals, and cervix and uterus, cervix in dog and horse, pig in the uterus. So these are the site of semen deposition. So after deposition in the female genital tract, these are the area where semens are deposited. So this is in female genital tract. So after deposition, the sperm have to reach to the oviduct, and the female genital tract, the environment is inhospitable to the spermatozoa because they have WBC or neutrophil infiltration, phagocytosis. There are presence of macrophages, so phagocytosis and inflammatory reaction occurs because spermatozoa is a foreign particle, and many of the spermatozoas are entrapped in the uterine fold. Therefore, they are destroyed due to phagocytosis. They cannot able to reach to the site of fertilization. So, at the site of deposition, millions of spermatozoa are deposited. Many of the spermatozoa are lost on the way of in the area of female genital tract. So, however, even though the female genital tract is inhospitable to spermatozoa, so many part of the female genital tract and they contribute for the survival and the transportation and the capacitation of the spermatozoa. So let's see what are the components in the female genital tract that survive, that helps in survivability of the spermatozoa in the female genital tract. So survivability in the female genital tract, that is cervix, one of the organs serving in the female genital tract. This area cervix, they have prominent ridge, mucosal crypts. They have extensive secretion where the spermatozoas are physically entrapped. And also another part, we have uterine horn oviduct junction. That junction where oviduct and the uterine horn and the oviductal junction. And also some said that there might be reservoir at the site of ampulla. So this reservoir play a very important role, and this reservoir keep the spermatozoa and they cause continuous slowly release of spermatozoa from this reservoir. And this reservoir also plays significant role to make the spermatozoa capacity for fertilization. And let's see transportations, how the female genital tract helps in the transportation of spermatozoa to the site of fertilization. So we know that folliculogenesis, the estrogen produced by the granulosa cell, this estrogen causes increased motility of the uterus. And not only it causes the motility of the uterus, but also estrogen prepare the receptor for oxytocin. So when the oxytocin is released from the pituitary, it acts upon the uterus, and estrogen develop the receptor in the uterus for oxytocin. So oxytocin and estrogen during this time they enhance the motility of the uterus. Therefore, the sperm movement in the female genital tract, the movement of the spermatozoa are helped by the motility of the uterus by these two hormones. And also the prostaglandins, which comes from the semen after deposition, after ejaculation to the female genital tract. This prostaglandins also helps and enhance the motility of spermatozoa and helps the movement of the spermatozoa. and also negative pressure of uterus, also somewhat contributing the transportation of the uterus. So I told you capacitation is the ability acquired by the spermatozoa for fertilization, which occurs in the female genital tract. So the ability of fertilization is acquired by the spermatozoa in the female genital tract. So the reservoir, it helps capacitation of spermatozoa, and also when the spermatozoa reached to the site of fertilization, they have to interact with the cumulus cell extracellular matrix of the cumulus cell. There are some component in the extracellular matrix in the cumulus cell. They also somewhat helps in the capacitation of spermatozoa, like acrosome reaction. So let's see what is the capacitation of spermatozoa, which is prerequisite for fertilization. Without capacitation, the sperm cannot able to fertilize. Let's see. It is simply the removal of removal of glycoprotein from the sperm surface. So glycoprotein is thought to be a protective layer for spermatozoa because it have to travel through the female genital tract, and a lot of fluid they have to interact. So therefore it is a protective layer. But this layer, we here we are telling that it is glycoprotein layer and or decapacitation layer. This layer prevent the spermatozoa to undergo capacitation. So while traveling in the female genital tract, they have to interact with lot of chemicals, molecule. So this layer is are destroyed or removed, and we have a capacitated spermatozoa. First, removal of the glycoprotein, and that occurs. Then, we have plasma membrane, and we have a acrosomal cap in the spermatozoa. This acrosomal cap, when they interact with the molecule, and this membrane, they undergo acrosome reaction. Acrosome reaction means uh, this membrane has broken, and there is exocytosis of the vesicle from acrosome. So acrosome have lot of vesicles which contain the proteolytic enzyme, and when this acrosome undergo reaction, this vesicle able to release to the surroundings by exocytosis, and this is called acrosomal reaction. So the membrane over the acrosome, it is fragmented, so that the vesicle which come out from the spermatozoa, and they can digest the tissue around the spermatozoa, because vesicle contain the proteolytic enzyme, that is acrosome reaction. So first, removal of the glycoprotein and the acrosome reaction is very important, and these are the prerequisite uh, for fertilization. So let's see this is structure. Uh, this is structure we have. These are cumulus cell, which is around the oocyte. After ovulation, this cumulus cell, that surrounds the oocyte, and between the cumulus cell we have this extracellular matrix. I have drawn as a net-like structure. This is extracellular matrix, and this matrix is main component is hyaluronic acid. So this hyaluronic acid, and there are a lot of component which gives the stability of the extracellular matrix between the cumulus cell. So when the sperm is capacitated, they remove the glycoprotein. They release the hydrolytic enzyme hyaluronidase. And when the spermatozoa undergo acrosome reaction, they release the proteolytic enzyme. And therefore, acrosin also one of the proteolytic enzyme which digests the acellular coating around the oocyte. So many of the proteolytic enzyme release from the acrosome, and those proteolytic enzyme digest this extracellular matrix, and the spermatozoa make a way to penetrate through the cumulus cell and reach to the oocyte. So let's see how the oocyte transported to the site of uh, fertilization, that is ampulla. After ovulation, this is the ovary. After ovulation, uh, this oocyte is released, and this oocyte is picked up by the infundibulum. This is a part of the oviduct. So at the when there is close to the ovulation, when at the time of ovulation, this infundibulum, this structure, they have a fembry. This fembry structure, they sweep over the ovary and they receive the oocyte, and this oocyte is uh, allowed to move toward the uh, ampulla site. And the oocyte, when it is received by the funnel shaped infundibulum, part of infundibulum, this oocyte is moving toward the ampulla, and this movement is caused by the ciliary movement of fembry. And also because oocyte are immotile, so they have to depend upon the ciliary movement of the oviductal cells over the oviductal cells, ciliary cells. Those cilia, they push the oocyte toward the ampulla. And also the contractile activity of those structure also helps to move the oocyte toward the uterus. Those ciliary movement and the contractile activity of the oviduct. So the prerequisite for fertilization for oocyte, it is very necessary that oocyte should complete its cytoplasm and nuclear maturation. And before fertilization, the oocyte have to complete its first meiosis division and release of first polar body. So this is our Zona pellucida, plasma membrane, and the oocyte, and the release of the first polar body to the perivitelline space. This space is called perivitelline space. So when they complete the nuclear division, they will release the first polar body to this space. And however, there is exception in some animals like dog and horse. These animals, they ovulate primary oocyte. Primary oocyte means they are not yet completed the first meiosis division at the time of ovulation. But in the oviduct, this stage is at dictyad stage. They have not completed the meiosis first division, or they have not released the first polar body. Therefore, before ovulation in the oviduct, this oocyte have to complete the first meiosis division. Then only they can enter to fertilization. Till that, the sperm have to wait for the oocyte to complete its first meiosis division. Even though in horse and dog they have released the primary oocyte, they have to complete it first meiosis division. Then only they can go for fertilization. The sperm have to wait till now. But very interestingly, in dog, after ovulation, they takes almost 48 hours time to complete its first meiosis division. Then only they go for fertilization. So therefore, the remaining time of the oocyte and the spermatozoa in the oviduct of beach or dog, it's quite longer than other domestic animals. So the lifespan, we have seen that after ovulation, the oocyte have to complete its first meiosis division. So, and the viability, how much time the oocyte can remain its fertilizing ability in the oviduct, how much time they can able to maintain their fertilizing ability before their destruction or before they're losing their ability for fertilization. So the viability of the oocyte is around 12 to 18 hours after ovulation in most domestic animals. This is a very short timing, 12 to 18 hours. So before this time, the oocyte have to be introduced by the spermatozoa for fertilization. Otherwise, there will not be any fertilization. But however, the sperm remains fertilizing ability for 24 to 48 hours in cow, ewe, and sow. So here, if we see the viability of the oocyte and the viability of the spermatozoa, so spermatozoa viability is quite longer than the oocyte, and also exceptional in mare, the spermatozoa can remain viable or having fertilizing ability in the female genital tract in horse, mare. It is around 120, 120 hours. Sorry, not days. It is 120 hours or 5 days. 5 days. But in spermatozoa in beach, in the female genital tract of dog, it's about 90 hours, almost 3 days. Because this mare and beach, they release the primary oocyte. They have to complete the first primary division, then only they can take part in the fertilization. So therefore, the sperm have to remain its fertilizing ability quite longer than in case of these animals. So when we see this ratio, spermatozoa almost remains its fertilizing ability almost twice than the oocyte viability. So that is very important because uh, the female receive the male or the spermatozoa before ovulation, so that the spermatozoa can travel to the site of fertilization and they can wait for the oocyte for ovulation. Because the oocyte viability is very short. So when the spermatozoa reach the site of fertilization before ovulation, the chances of fertilizing capacity or percentage of fertilization is much better. So sexual receptivity, we have seen the viability of oocyte is uh, less than viability of the spermatozoa in the female genital tract. Therefore, the sexual receptivity in most of the domestic animal, that is estrus, they receive the male. It is the time sexual receptivity. It is the time period where female receives male for copulation or mating, and this mating occurs at least 24 hours before ovulation. This is very important, before ovulation for successful fertilization. So before it gets ovulated, the animal they receives the male for copulation or mating. However, it also seen in induced ovulator. Example, cat. Now these animals, when the penis touches the vagina, then only they undergo ovulation. So this induced ovulator also, the duration from copulation to ovulation is about 24 hours or sometimes more. So that area, so before ovulation, the purpose is that before ovulation, the spermatozoa have to acquire its fertilizing ability and reach to the site of uh, fertilization. So different animal, the time of ovulation is given here. Mare 1 to 2 days before the end of estrus. Estrus is the time where it shows the sexual receptivity, then where the female receives the male. So this is the time of ovulation in different domestic animals. So we have discussed that at the time of fertilization, we discussed that how the sperm they reach to the site of fertilization, how the female genital tract they helps the spermatozoa to reach to the site of fertilization, and the viability of the spermatozoa and the viability of the oocyte and the prerequisite of oocyte and the sperm. So this capacitation is very important for sperm. That is removal of glycoprotein, acrosome reaction, and hydrolytic enzyme release and proteolytic enzyme release. So these are the enzymes. They release and they digest the extracellular matrix and sperm able to go through the cumulus cell and reach to the site of reach to the close to the or reached to oocyte. So this capacitated spermatozoa, they release these enzymes and they digest the extracellular matrix and they are moving toward the oocyte. So as they move toward the oocyte, when they touch the membrane of the oocyte, that is zona pellucida, this membrane, zona pellucida, after penetrating the after coming through the cumulus layer. So as they enter to the zona pellucida, as soon as it touches the membrane or enters the zona pellucida, no, there is some modification occurs in the zona pellucida. And that modification in the zona pellucida is called Zona reaction. So as soon as the sperm touches the or penetrate the zona pellucida, so the membrane of the zona pellucida undergo some modification and they prevent the entry of other sperm. So that is called zona reaction. So when one sperm is penetrated into the zona, other sperm they cannot penetrate. This membrane undergoes some modification. Let's see how this modification occurs. So this is the oocyte. In the cortex part of the oocyte, this part of the oocyte, this area, we have granules. This secretory granules. These secretory granules are also called cortical granules. This cortical granule, they have the component or enzymes or some material they release to the zona layer. This release to the this space and they move and fuse with the zona layer and they do some modification over this layer, and because of that cortical granule which is released by exocytosis and they fuse with the zona layer, they do the modification of this layer where they cannot interact with other sperm or they prevent the entry of other sperm, and that modification leads to impermeable to sperm or prevent polyspermy. That is called zona block, and because of the cortical granule they are released by exocytosis and the component which is in the granule they fuse with the or cause some modification over the zona layer and they cause the block or we call it zona block to prevent the polyspermy. So when the sperm is successfully able to penetrate and other sperms are blocked, when only one sperm is able to penetrate the zona, and the head portion of the spermatozoa, not the tail portion, but the head portion of the spermatozoa, the membrane of the spermatozoa, they fuse with the membrane of oocyte. That is ulema, plasma membrane of oocyte. That plasma membrane of spermatozoa are fused with the plasma membrane of oocyte, and the spermatozoa they release their genetic material and other component to the oocyte or cytoplasm. So not only genetic material but other component also they have released to the released to the oocyte cytoplasm, or mainly I will be talking about here genetic material. So that is oocyte genetic material, and that is the sperm they have given its genetic material. So when the genetic material has released or to the cytoplasm of the oocyte, and they form the pronuclear, male pronuclear and the female pronuclear, the pronuclear formation. the that pronuclear formation occurs. And when these pronuclei come together and they fuse together, they form zygote, one cell embryo, that is single cell embryo. When the female genetic material and the male genetic material, they the pronuclei, they fuse together, and all the chromosome from the male part and the female part, they uh, they align each other, and they form the zygote, and that is single cell zygote, and that is the embryo, single cell embryo has formed after fertilization. So after fertilization, the zygote undergo cleavage. Cleavage means division of the embryo. They go 2 cell, 4 cell, and they divide, and they form the morula, compact morula. The cell divide within this zona layer. The cell divide itself, and they become many cells. And after morula, we'll be not talking about the embryogenesis detail. The blastocyst structure. Some of the cells, they form inner cell mass. These are inner cell mass, and this part we call it trophoblast. They form this surrounding cells. They form the placenta. So this structure we can find because there is formation of the cyst. We call it blastocyst. That structure has formed during embryogenesis. So when the zygote has formed, the zygote remain in the oviduct and they go on embryogenesis, division, or cleavage. So that is very important to remain the zygote or the initial part of embryogenesis in the oviduct. So during that period, the uterus, during the ejaculation, when sperm moves to the uterus before fertilization, there is inflammatory response, and lot of sperms in the uterus, lot of sperm in the uterus have to remove before embryo reach to the uterus. So during embryogenesis or during fertilization, after fertilization, when the zygote has formed, the zygote remain in the oviduct almost 4 to 5 days, and this zygote undergo cleavage within the oviduct. So before they comes to the uterus have to remove all the spermatozoa from the uterus or destroyed by macrophages, and also the uterus have to secrete sufficient amount of uterine milk, and that milk is very important for the embryo development under the influence of progesterone. So, and also uterine milk is necessary for development of embryo. And very significant one, before folliculogenesis, after during folliculogenesis, the whole system is dominated by the estrogen. But after ovulation, the structure form the corpus luteum, and those corpus luteum, they produce the progesterone, and now it is become progesterone dominant. So during estrogen dominant, they cause uterine motility. So before ovulation, they cause uterine motility, help in the sperm transportation, and that uterine motility is unfavorable now for embryo. But when the system become progesterone dominant, this progesterone prevent the uterine motility, and when the uterus become prevented for its motility, and it is suitable for embryo for implantation. So motility of the uterus have to be prevented, and that is prevented by progesterone. So this is our blastocyst. When it is reached to the uterus, this blastocyst have to be hatched from they have to be come out from the zona pellucida. They have to come out from the zona pellucida, and that structure, that process is called hatching blastocyst. And this blastocyst is going to penetrate the endometrium or implantation of the endometrium. and this superficial layer it will form the placenta. So let's see what component this blastocyst, this hatched blastocyst is secreting to the uterus and implantation. Let's say the mechanism of implantation in ruminant. So in ruminant, the blastocyst, hatched blastocyst, they secrete one very important compound, protein, that is interferon tau. This interferon tau block the receptor for block the estrogen and oxytocin receptor. And while blocking the estrogen and oxytocin receptor, they prevent the motility of uterus. And when the motility of uterus is prevented, the PGF2 alpha, which is secreted from the uterus, when these estrogen and oxytocin receptors are blocked and the motility of the uterus is almost blocked, then prostaglandins they cannot secrete from the uterus. They don't secrete because prostaglandins they cause luteolity and it destroy the corpus luteum. When corpus luteum is destroyed, progesterone secretion stops, and progesterone is very important for maintenance of pregnancy. So prevention of PGF2 alpha is very important in ruminant, in cow and cow especially, and sheep. That's very important for the prevention of PGF2 alpha. And interferon tau cause the stopping release of PGF2 alpha by blocking estrogen oxytocin receptor. And while blocking this PGF2 alpha, corpus luteum maintained and progesterone production is maintained. When progesterone production is maintained, maintenance of pregnancy is maintained. That's how it occurs for the maintenance of pregnancy. So here, estrogen also is said to play a very important role. Estrogen secreted by the embryo, and this estrogen secreted by the embryo, they also said to inform the uterus that there is a presence of embryo. So the animal system, they have to recognize the presence of embryo in the genital tract. Even some author has said that when the embryo has formed in the oviduct, that time only some species can able to recognize that there is a presence of embryo. But that presence of embryo is not enough for maintenance of pregnancy. That embryo have to implant it, have to penetrate the uterus for implantation. And when it is implanted, they secrete some component. In ruminant, it is interferon tau, and that component helps to maintain the pregnancy. So it is different in horse, mare. That is, we'll be talking about hormones of pregnancy in next video. But some animals they depend upon corpus luteum, and some domestic animals they depend upon placenta completely. So in ruminant, interferon tau is one of the very significant for recognition or an establishment of a pregnancy and implantation. So in mare, the movement of the embryo in the genital tract is important for establishment of pregnancy. And transuterine migration of embryo in litter-bearing animals are very important for establishment of pregnancy and recognition of embryo. In pigs, the requirement is 4 embryo for the establishment of pregnancy, and this embryo also move intrauterine migration occurs in this species. The embryo, when they comes to the uterus, they have to depend upon its yolk and the uterine milk until it fixes with uterine endometrium. On an average, 2 to 5 weeks is required after fertilization for implantation. In cat, 2 weeks, and cattle 5 weeks to completely depend upon the endometrium. for their nutrient supply. Okay, thank you. Next video we'll be discussing some of the hormones which helps for maintenance of pregnancy.